So hi, it's me again. And now, actually a very similar topic, so hopefully you were paying attention to the last talk because you need that to understand this one. But now we're going to talk a little bit about inflammation and another process called cellular senescence. And they're very similar because, and we'll get to that in a second, but basically what I want to present to you here is some additional strategies that we could use to address the pro-inflammatory environment. These are my disclosures, nothing relevant to this particular talk. So I just showed you this data from Ralph's group that is just reiterating the fact that aged mice have delayed healing and that macrophages contribute to that delayed healing. Same data, different format. But one of the things that is problematic in preclinical studies is what I didn't tell you earlier is that to get an aged mouse, the NIH's definition of aged mouse is 24 months. So this is a really time and cost extensive process to get a quote-unquote old mice. So what we've been trying to develop is a better way, a better model that might not take quite so much cost. And just to give you perspective, an old mouse in the NIH through the, if you wanted to buy it, is about $350 per old mouse, or waiting 24 months. So we were really interested in trying to develop a system, a model system that could replicate the age-related delay we saw in the last talk without having to wait so long or cost so much money. And the reason we want to do this is exactly what I told you earlier, is that we have this ambition to try to test novel therapeutics, and we want to be able to do this in a more affordable, rapid manner. So this is a progeria, Hutchinson-Gilford progeria system that you're all familiar with, and it turns out there's actually a mouse mutation that we can, a genetic mutation to laminin A that can replicate this disease in a mouse model. And so we have previously worked with this mouse model to publish that it is more or less osteoporotic, it's sarcopenic, and it has a fragile skeleton. So this first paper that we published a while ago just showed that these mice, they're old, they die at about 3 months of age, which is much more close to a 24-month-old mouse, and they have reduced bone mineral density in their skeleton. And the question was, do these mice also heal their fracture more slowly? So a lot of data here that basically shows that these mice have delayed healing. So on the top is showing that at day 14, there's much more cartilage and much less bone in these Z24 or progeric mice. That's consistent at day 21. And really, we haven't ever seen these fractures go on to heal in a mouse model. We also show that mechanically their bones are much less weak and their fractures are also consequently much weaker. But we wanted to then build off of the last data that suggests that macrophages are the leading cause of this immune dysregulation in an old mouse, and now we have new tools and we can do this spectral flow immunophenotyping, where we look at all of the different myeloid and lymphoid cells and can really try to understand at a much broader level what cells are dysregulated. So this is the complex panel. It was done over at the Buck Institute over in Marin, who has this really incredible core facility to be able to do this spectral flow immunophenotyping. And so what we saw is that we generally saw that these mice have a dysregulated immune system where they have increased myelopoiesis and diminished lymphopoiesis pretty quickly after the fracture, at 3 days after the fracture. And so you can see that in a number of different measures, but what we were really interested about is not only is there a macrophage dysfunction, but we're now starting to see that T cells and B cells, granulocytes, a number of different other cells are also dysregulated. And I think the literature is just slowly catching up for us to start thinking about a role of the adaptive immune system in addition to the innate immune system in fracture healing. So this is at 3 days post-fracture, and then I just put some sample data on the right-hand side that shows that they're dysregulated through the whole time course of healing. And so this is something that, again, goes into this idea of immune dysregulation with age, and now we're seeing it in this model of accelerated healing. So in addition to this immune system dysregulation, we wanted to start thinking about the role for cellular senescence. So cellular senescence is the process of basically cellular aging, and it's caused by a number of things: DNA mutations, telomere shortening, and what happens with these senescent cells is they start to secrete pro-inflammatory factors such as TNF-alpha, IL-1 beta, a number of other things, but they also are cells that are non-replicating, so and are resistant to apoptosis. And what that means is these cells, if we look at if the kind of red cell indicates a senescent cell, these cells are basically poisoning all the healthy cells around them, and because they're not dying or replicating, they're really hard to target, and so these live in your body and accumulate as we get older. And now there's a number of diseases that have been associated with cellular senescence, including osteoporosis, osteoarthritis, sarcopenia, rheumatoid arthritis. And it really is very understudied in the world of fracture healing. So we wanted to start to understand and measure cellular senescence. And so developed a protocol to basically be able to take blood and run it through flow cytometry and looking for a cellular signature of beta-galactosidase activity, which I forgot to mention in the last slide is a hallmark of cellular senescence. We're able to measure that, and now we can assay both humans and mice for the number of senescent cells they have circulating in the blood or bone marrow or lymphoid processes. So this is a paper that was just accepted in Aging Cell, and it has about 200 patients. And you can see here with this assay, we can measure an increase in senescence with chronological age. Interestingly, we also see that males tend to have more senescence than women, proving the point that we really are the stronger sex. And we can measure this in humans, and we also see it in this mouse model we have. So this is just showing that these accelerated aging modeled mice have increased cellular senescence compared to wild type, and they also have the increased immune pro-inflammatory phenotype that we saw earlier. So they have increased IL-1 beta, increased TNF-alpha. So this helps to indicate, just like the aged mouse model, why these mice might have delayed fracture healing. And this is just showing that they have an increased variation. I'm going to skip over that for time. So where this becomes part of the innovation is now we can characterize that there's immune dysfunction, and also cellular senescence is a factor contributing to aging in humans and in mice. And we can start to test senolytics or therapeutics that target senescent cells. And so this is just, again, this idea that senescence starts to build up systemically in your body and all the different organs as you age. And now can we identify drugs that can target those senescent cells? Because since they aren't replicating, we need special drugs. And there's a lot that have been explored, but tend to be drugs that are chemotherapeutics, and so they can be very toxic. Dasatinib, quercetin, these are many of the drugs people have tested as senolytics. We have been interested in testing a drug, actually it's a supplement called fisetin, and it acts at this part of the cell to inhibit a number of cellular processes. And so it specifically goes in and gets rid of the senescent cells, and this is an example from 10 patients that we've seen in the clinic and given them fisetin to show that we can reduce measurements of senescent cells, both in their blood using that same assay I talked about earlier and other pro-inflammatory markers. So this has been used at the Stedman Clinic as a clinical trial just generally to address aging. And we were curious if we could now start to look at it as a therapeutic to address age-related delay in mice or in fracture healing. So we gave those progeric mice the drug fisetin, and we can see that we can actually decrease their bone mineral density loss by giving this supplement. And so that was one of the first things that was encouraging to us. Not only did we improve their bone mineral density, but we actually decreased the amount of IL-6 in their blood, so decreased the pro-inflammatory microenvironment of these mice. We also gave it to mice after we fractured their bone, and specifically we gave it at 2 different times. We gave it early at day 2, which is around the peak of the pro-inflammatory time, and then we gave it later. And the reason we did it later is in fracture healing, the pro-inflammatory response is really key. So we wanted to understand what was the time course of when to give this drug. And this is an experiment that was funded by the OTA, a research grant, and we're really excited here because we can see that the fisetin appears to improve fracture healing and decrease the pro-inflammatory environment. And we're still increasing our N here, but we are interested in exploring this in a mouse model so that we can maybe go into humans and think about this as a possible way to attenuate age-related delays in fracture healing. So from bench to bedside, I think there's a lot of value in simultaneously doing human and mice experiments to try to test in an observational study in humans what's happening, and then you can therapeutically intervene in mice to test possible new strategies.